equals 1/2 t plus 1/2 t by 1/2 t plus 1/2 t equals 1/4 tt plus 1/2 tt plus 1/4 tt. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarfs was homozygous TT. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using mathematical probability, by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has TT or TT composition, cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism P plants here, showing a dominant phenotype and whose genotype is to be determined, is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 5.5 shows the results of typical test cross where violet color flower W is dominant over white color flower W. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition for a test cross? Figure 5.5 diagrammatic representation of a test cross. Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today these rules are called the principles or laws of inheritance, the first law or law of dominance and the second law or law of segregation. 5.2.1 Law of Dominance. I. Characters are controlled by discrete units called factors. E. Factors occur in pairs. E. In a dissimilar pair of factors, one member of the pair dominates dominant, the other recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 5.2.2 Law of Segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar while a heterozygous one produces two kinds of gametes each having one allele with equal proportion. 5.2.2.1 Incomplete Dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, snapdragon or Antirrhinum sp. is a good example to understand incomplete dominance. In a cross between true breeding red-flowered RR and true breeding white-flowered plants RR, the F1 RR was pink, figure 5.6. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 RR red, 2 RR pink, 1 RR white. Here the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1 dominant, recessive ratio. What happened was that R was not completely dominant over R, and this made it possible to distinguish RR as pink from RR red and RR white. Figure 5.6 Results of monohybrid cross in the plant snapdragon, where one allele is incompletely dominant over the other allele. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, i.e., as a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on, and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for production of I. The normal, less efficient enzyme, or E. A non-functional enzyme, or E. No enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, i.e., it will produce the same phenotype trait, i.e., result in the transformation of substrate as such equivalent allele pairs are very common. But, if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype as the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Table 5.2, table showing the genetic basis of blood groups in human population. Allele from parent 1. Allele from parent 2. Genotype of offspring. Blood types of offspring: IA, IA, IAIA, A, IA, IB, IAIB, AB, IA, I, IAI, A, IB, IA, IAIB, AB, IB, IB, IBIB, B, IB, I, IBI, B, I, E, O. 5.2.2.2 Codominance. Till now we were discussing crosses where the F1 resembled either of the two parents' dominance or was in between, incomplete dominance. But, in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles IA, IB and I. The alleles IA and IB produce a slightly different form of the sugar, while allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over I. In other words, when IA and I are present, only IA expresses because I does not produce any sugar, and when IB and I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore, a total of 6 different genotypes of the human ABO blood types. Table 5.2. How many phenotypes are possible? Table 5.2, table showing the genetic basis of blood groups in human population. Allele from parent 1. Allele from parent 2. Genotype of offspring. Blood types of offspring: IA, IA, IAIA, A, IA, IB, IAIB, AB, IA, I, IAI, A, IB, IA, IAIB, AB. IB, IB, IBIB, B, IB, I, IBI, B, I, E, O. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, i.e., 3 alleles, governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles B and B. Starch is synthesized effectively by BB homozygotes, and therefore, large starch grains are produced. In contrast, BB homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, BB seeds are round and the BB seeds are wrinkled. Heterozygotes produce round seeds, and so B seems to be the dominant allele. But, the starch grains produced are of intermediate size in B-flat seeds. So if starch grain size is considered as the phenotype, then from this angle, the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. 5.3 Inheritance of 2 genes. Mendel also worked with and crossed pea plants that differed in 2 characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape. Figure 5.7. Mendel found that the seeds resulting from the crossing of the parents had yellow-colored and round-shaped seeds. Here can you tell which of the characters in the pair's yellow-green color and round wrinkled shape was dominant? Figure 5.7 results of a dihybrid cross where the two parents differed in two pairs of contrasting traits, seed color and seed shape. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols Y for dominant yellow seed color and Y for recessive green seed color, R for round-shaped seeds and R for wrinkled seed shape. The genotype of the parents can then be written as RRYY and RRYY. The cross between the two plants can be written down as in figure 5.7 showing the genotypes of the parent plants. The gametes re and reunite on fertilization to produce the F1 hybrid RRYY. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 5.3.1 Law of Independent Assortment. In the dihybrid cross figure 5.7, the phenotypes round, yellow, wrinkled, yellow, round, green and wrinkled, green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green, with 3 round, 1 wrinkled. This derivation can be written as follows. 3 round, 1 wrinkled, 3 yellow, 1 green equals 9 round, yellow, 3 wrinkled, yellow, 3 round, green, 1 wrinkled, green. Based upon such observations on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that, when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen in the F1 RRYY plant. Consider the segregation of one pair of genes R and R. 50% of the gametes have the gene R and the other 50% have R. Now, besides each gamete having either R or R, it should also have the allele Y or Y. The important thing to remember here is that segregation of 50% R and 50% R is independent from the segregation of 50% Y and 50% Y. Therefore, 50% of the R-bearing gametes has Y and the other 50% has Y. Similarly, 50% of the R-bearing gametes has Y and the other 50% has Y. Thus there are 4 genotypes of gametes, 4 types of pollen and 4 types of eggs. The 4 types are Re, and Re each with a frequency of 25% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the two sides of a Punnett square, it is very easy to derive the composition of the zygotes that give rise to the F2 plants figure 5.7. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? S. No. Genotypes found in F2. Their expected phenotypes. 5.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons, it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of genes or factors, in Mendel's words, as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other, was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors, genes, were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, 3 scientists, de Vries, Correns and von Schermuck, independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appear to double and divide just before each cell division. These were called chromosomes, colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement figure 5.8 to explain Mendel's laws table 5.3. Recall that you have studied the behavior of chromosomes during mitosis, equational division, and during meiosis, reduction division. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. Figure 5.8 Meiosis and germ cell formation in a cell with 4 chromosomes. Can you see how chromosomes segregate when germ cells are formed? Table 5.3 A comparison between the behavior of chromosomes and genes. A. B. Occur in pairs. Occur in pairs. Segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Segregate at gamete formation and only one of each pair is transmitted to a gamete. Independent pairs segregate independently of each other. One pair segregates independently of another pair. Can you tell which of these columns A or B represent the chromosome and which represents the gene? How did you decide? During anaphase of meiosis, the two chromosome pairs can align at the metaphase plate independently of each other. Figure 5.9. To understand this, compare the chromosomes of 4 different color in the left and right columns. In the left column, possibility I, orange and green as segregating together. But in the right-hand column, possibility 2, the orange chromosome is segregating with the red chromosomes. Figure 5.9 Independent assortment of chromosomes. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster figure 5.10, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes, the male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low-power microscopes. A. B. Figure 5.10 Drosophila melanogaster A. Male B. Female. 5.3.3 Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome, section 5.4, and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination figure 5.11, cross A, while others were loosely linked, showed high recombination figure 5.11, cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of the Human Genome Sequencing Project, described later. Figure 5.11 Linkage, results of two dihybrid crosses conducted by Morgan. Cross A shows crossing between gene Y and W, cross B shows crossing between genes W and M. 5.4 Polygenic Inheritance. Mendel's studies mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans we don't just have tall or short people as two distinct alternatives but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, i.e., the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control skin color in human, with the dominant forms A, B and C responsible for dark skin color and the recessive forms A, B and C for light skin color. The genotype with all the dominant alleles AABBCC will have the darkest skin color, and that with all the recessive alleles AABBCC will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. 5.5 Diatrippy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a Pliotropic gene. The underlying mechanism of Pliotropic in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. 5.6 Sex determination. The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic chromosomal basis of sex determination. Henking, 1891, could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Henking gave a name to this structure as the X-body, but he could not explain its significance. Further investigations by other scientists led to the conclusion that the X-body of Henking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects the mechanism of sex determination is of the exotype, i.e., all eggs bear an additional X chromosome besides the other chromosomes, autosomes. On the other hand, some of the sperms bear the X chromosome whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Among the males an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY, while female have autosomes plus XX. In human beings and in Drosophila the males have one X and one Y chromosome, whereas females have a pair of X chromosomes besides autosomes, figure 5.12a, b. In the above description you have studied about two types of sex-determining mechanisms, i.e., exotype and XY type. But in both cases males produce two different types of gametes, A, either with or without X chromosome, or B, some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanism is designated to be the example of male heterogamety. In some other organisms, e.g., birds, a different mechanism of sex determination is observed, figure 5.12c. In this case the total number of chromosome is same in both males and females. But two different types of gametes in terms of the sex chromosomes are produced by females, i.e., female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In these organisms the females have one Z and one W chromosome, whereas males have a pair of Z chromosomes besides the autosomes. Figure 5.12 Determination of sex by chromosomal differences, A, B. Both in humans and in Drosophila, the female has a pair of XX chromosomes, homogametic, and the male XY, heterogametic composition. C. In many birds, female has a pair of dissimilar chromosomes ZW and male two similar ZZ chromosomes. 5.6.1 Sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females, these are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of an X and Y chromosome are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. 5.6.2 Sex determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from the union of a sperm and an egg develops as a female queen or worker, and an unfertilized egg develops as a male grown by means of parthenogenesis. This means that the males have half the number of chromosomes than that of a female. The females are diploid having 32 chromosomes and males are haploid, i.e., having 16 chromosomes. This is called as Haplodeploid sex determination system and has special characteristic features such as the males produce sperms by mitosis. Figure 5.13 They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. Figure 5.13 Sex determination in honeybee. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? 5.7 Mutation. Mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in Chapter 6, one DNA helix runs continuously from one end to the other in each chromatid, in a highly supercoiled form. Therefore, loss, deletions, or gain, insertion, duplication of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arises due to change in a single base pair of DNA. This is known as point mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes frame shift mutations, see Chapter 6. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutations in organisms, it is a mutagen. 5.8 Genetic disorders. 5.8.1 Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in pea plant or some other organisms are not possible in case of human beings, study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in a several of generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool, which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis have been shown in Figure 5.13. Figure 5.13 Symbols used in the human pedigree analysis. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do take place occasionally. Such an alteration or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. 5.8.2 Mendelian Disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in Figure 5.14 for dominant and recessive traits. Discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosome. Figure 5.14 Representative pedigree analysis of A. Autosomal dominant trait, for example, myotonic dystrophy. B. Autosomal recessive trait, for example, sickle cell anemia. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. The daughter will not normally be colorblind, unless her mother is a carrier and her father is colorblind. Hemophilia, the sex-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female carrier for hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for the gene or heterozygous. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS show the disease phenotype. Heterozygous HBA/HBS individuals appear apparently unaffected, but they are carrier of the disease as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. Figure 5.15. The defect is caused by the substitution of glutamic acid (GLU) by valine val at the sixth position of the beta-globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of the beta-globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Figure 5.15. Figure 5.15 Micrograph of the red blood cells and the amino acid composition of the relevant portion of beta chain of hemoglobin, A. From a normal individual, B. From an individual with sickle cell anemia. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also excreted through urine because of its poor absorption by kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, A and B chains, that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules, resulting into anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In a thalassemia, production of a globin chain is affected, while in B thalassemia, production of B globin chain is affected. A thalassemia is controlled by two closely linked genes HbA1 and HbA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecules produced. While B thalassemia is controlled by a single gene HBB on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. 5.8.3 Chromosomal disorders. The chromosomal disorders, on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosomes, called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner's syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 4623 pairs. Out of these, 22 pairs are autosomes and 1 pair of chromosomes are sex chromosome. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome, respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Kleenefelter syndrome are common examples of chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, trisomy of 21. This disorder was first described by Langdon Down, 1866. The affected individual is short-statured with small round head, furrowed tongue and partially open mouth, figure 5.16. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Figure 5.16, a representative figure showing an individual inflicted with Down syndrome and the corresponding chromosomes of the individual. Kleenefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype of 47, XXY. Such an individual has overall masculine development, however, the feminine development of breast, i.e., gynecomastia, is also expressed, figure 5.17a. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosomes, i.e., 45 with X0. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters, figure 5.17b. Figure 5.17 diagrammatic representation of genetic disorders due to sex chromosome composition in humans. A. Kleenefelter syndrome. B. Turner syndrome.